Everyone, my name is, actually, I'm going to shut the door.
This is a full question. That's not a question. Yeah.
My name's Tracy McGinnity. I work in Wilmot's Shared Resources in the management, and we're hosting this Lunch and Learn seminar series. We get support from Vivian Palladoro, a retired URMC clinical pathology administrator, and she's kind of our shared resource champion and helps us with financial support to provide this series. Just some quick announcements. I wanted to let you all know of the schedule for the remainder of the year for the series. Again, it takes place on the second Tuesday of every month. The remaining seminars will be held in this room. The first half of the year, we were in a different auditorium. So today we have our spectral cytometry presentation. In September, we have our biobank shared resource presentation, followed by metabolomics in October, and biostatistics and bioinformatics in November. December, I'm not sure if we will be hosting a general shared resource lunch and learn, or if we will be hosting a community-focused event. So it's still to be determined. Another announcement is that we have our annual Community Cancer Action Council retreat. If you want to present a poster, they are seeking more basic science research-focused projects. And it's a good opportunity to present to more of a lay audience. So if you want to practice communicating science to those that don't have the technical expertise that you do or the knowledge of your project at such a high level, this is a great opportunity to present your research. It's a fun day, and there's a lot of the community working groups and champions that are present. So abstracts are due Friday, September 5th, and the actual retreat is on Friday, October 17th. And you don't need to have an abstract in order to attend. So if you just want to attend, that is an option as well. Final announcement is that we are now accepting submissions for our 2026 shared resource calendar. So if you had a paper published or a paper in progress and you have some really cool images that you took for your research, feel free to submit them. We will review them. And then the calendars will be ready by the Wilmot Symposium in November. And this year, we're trying to focus our calendar more on NCI designation efforts. So we may have fewer images selected, but we still want to kind of, I don't know exactly the word, just feather them in throughout the calendar. So those are my announcements. I'm happy to introduce members of the Cytometry Shared Resource team who will be presenting more in depth on spectral cytometry today. So I will let Matt speak first.
Yeah. All right. Thanks everybody for coming. I don't have like an overview introduction slide, but I will briefly say a few words and then I'll dive in. So I'm going to present today with, along with Tim Mosmann and Nate Laniewski, about full spectrum or spectral cytometry. I'm going to start with kind of an overview and an introduction to our facility and what options we have. Tim's going to dive into some data and go a little bit deeper there, and Nate's going to do the same. And we all have come from different kind of experiences with the technology, and our intention is to leave a lot of time for questions. So please sprinkle them in throughout the talks if you want to, and we want to leave some time for questions afterwards, because while we've had the technology here in Rochester for about 5 years, it's still fairly new, and there's a lot of people that are just getting exposed to it. So a dialogue around that can be useful. So that's kind of the second half of this, or the last third. We want to leave open for communication and conversation and comparisons between the different tools that we have and whatnot, right? Rather than feed it all to you, we want to hear your thoughts and get some information back from you guys as well. So I am going to start again with the overview stuff. This is excerpted from a longer talk that I've given in the past. By the way, on that note, if this strikes your fancy and you want to learn more or you want to dive a little bit deeper, I'm happy to do things like join you in a lab meeting and have that conversation, or whatever your preference. Because I know a lot of times that you hear a lot of things, but it doesn't internalize as well in a session like this. So if you want more information, again, please do always come back. So we are part of the Cytometry Shared Resource or the Flow Cytometry Resource, depending on which side of the building you're coming from. You know, we got a great team. Myself and Tim Mosmann are going to talk today. Nate's not part of the crew, but he's been working with us for a long time at this point.
He's into remote.
Oh, yeah. Sorry. Thank you. Is this going to work? Is it going to? Hey, look at that. All right. I'm not going to introduce everybody or anything like that because I don't have a ton of time. But please do come see us if you want to dive a little bit deeper in and have a longer chat. Who's not on the slide, Ravi Misra has joined the team. Sorry, I don't have the updated slide with his picture and everything. But Ravi has joined the team as a co-director as well, or as an associate director as well. Tim Bushnell is moving out of the role and has more of an administrative role in cart and stuff. So Ravi's coming in. So you'll hear a lot more from him over the future. Okay, so quick look at what we have available to our users. There are two Cytec Aurora full spectrum analyzers. They are both 5 lasers and have 64 detectors. If you come from traditional flow, that doesn't mean they're 64 color instruments. That's not how full spectrum works, but they are larger quantity, larger quantity, target number instruments. So I'll dive into how that works in just one second. There is also an Aurora CS, a Cytec Aurora CS cell sorter. That's what CS stands for. It's, again, a 5 laser configuration. They are all matched, which, again, I will refer to a bit more in the future. And that has 6-way sorting and indexing and all kinds of stuff. So it is an excellent sorter with all the full spectrum benefits, which, as far as I'm concerned, is a huge, huge perk. The newest thing that I'm going to introduce today and maybe talk a bit more about towards the end, but not going to go super far today, is the newest machine, the Fax Discover A8. I just sent an announcement email about this recently. It has been installed. It is in use a little bit already. This is, again, a 5 laser, same 5 lasers, 5 laser 78 detector full spectrum instrument that also has 3 blue laser specific imaging channels as well. It's not a CCD camera-based imaging technology. It's a different thing that I'm not going to dive into today, but we can do some imaging with that tool as well. Whether it's just bright field or a bright field-like image, or you actually want to get into some fluorescence imaging, we can do some of that on this machine. So we're just scratching the surface of the capabilities here, and I'm happy to talk a little bit about how that works in the future, or towards the end here. But it is the newest, coolest tool in the lab, and it also looks pretty sharp. They did a good job with the styling this time around. Remember the old LSR2s were not very well styled, so they've come a long way in that respect as well. So full spectrum cytometry. The technology that we have here was introduced by Cytec in 2017 and kind of took off. It actually has been around longer than that. I'm not going to go back through the history and stuff, but really when Cytec put this machine out in 2017, it just took off, and it's been huge ever since. The idea here is that instead of with traditional flow, where you pick a detector on the machine and pick a fluorochrome that will fit that detector, this machine doesn't work that way. Instead of measuring each fluorescence fluorochrome by detector, it measures the entire spectrum, hence the term full spectrum, and you get to see an entire fingerprint or signature or whatever term you want to use for that fluorochrome. So PE in our traditional machines was measured off of the blue or the yellow-green laser and then captured about 580 or so, right? In the full spectrum instrument, we still have that peak yellow-green signal here, but it's actually measured in every one of the lasers and every one of the detectors. And what the system then uses is that spectral fingerprint in order to unmix it and to identify what fluorochrome is present. So any given cell has a pattern of fluorescence on it, and you can read that pattern of fluorescence and you can make sense of the data. So because there's so much more information, one, the computer processing, all that kind of stuff, and as Tim's going to get into, there's a very fine margin for how all of this works. But with that much information, you can make a finer determination of what fluorescence is present, and that's how we can get more fluorochromes and different combinations of fluorochromes that we couldn't evaluate before. Again, without diving too deep on that, the first thing that most people recognize as the benefit of full spectrum is more fluorochromes, right? Personally, I think that is great, but it's actually secondary for me in a lot of ways. There's 3 specific things I want to mention that allow that I think are even larger separation there. One is the longitudinal stability that the machines have. They have extremely good engineered stability, physical stability, but they also have a really good method of normalizing the data over time as well. And those two things together give you really good longitudinal stability. The other thing is standardized configurations. I said before that we have 3 5-laser Cytec Aurora instruments, one of them being a cell sorter. They all have matched underlying hardware specifics, which allows us to generate experiments on one machine and then transfer that experiment to another machine and open it up and then continue on with what you're doing. Nothing is perfect. I don't want to give you the impression that everything will open up and be exactly the same, but you can open them up and go back and forth. Again, if you're used to the traditional instruments, especially the FACS-DIVA cytometers that we have around, we have a number of matched instruments, but you can never go back and forth quite like that. You have to rebuild it on another machine and you have to start from zero. That's not true here. It gives us much more flexibility that we really appreciate. A couple of weaknesses. Again, it's kind of on the new side, so there's a little bit of learning curve, especially with regards to the theory and how you pick fluorochromes and stuff. Record keeping and annotation of data is extremely important here, just because of, again, the longitudinal stability and how we're keeping track of things. There's not as much of a separation for one-off projects as there are for long-term projects and for bigger things. You could still do one-off projects on these machines, but you lose a little bit of that benefit, especially with the longitudinal stability and stuff. Again, it's just a little bit deeper on what the matched configurations can do for you. First thing is with respect to building experiments, there are a number of ways you can do this at the machine in real time, which is perfectly fine, but you can also do it in advance in the cloud or in advance on one of our satellite computers as well. It also means if you have colleagues at a different institution that are using the same type of machine, you can save templates and share them with them. They can reopen them without having to worry about anything. Much more fluid, much more flexible in terms of sharing there. They also, because of this match configuration, I think Tim's going to allude to this a little bit more, is have a very good way of predicting what the fluorochromes will look like on each machine because of that matched underlying configuration. So you can predict things like similarity, how similar the fluorochromes are going to be. You can predict things like the complexity of the experiment they're trying to do. I'm not going to dive into what those numbers mean today, but I just want to introduce them. It does allow you to kind of predict a lot of things in advance better than we could on the other machines. Also allows for standardized panels. So there's a number of pre-made panel kits you could purchase if you wanted to. Many people like to kind of sprinkle in their own flavors in there and their own targets. But if you wanted to purchase one of those pre-made kits, they are really nice in that it comes with all the pieces you need. It even comes with an experiment template that you don't even have to create. You just plug it into the software and it'll open up and you can just start running your experiment. So again, this match configuration, I think for me, is a huge benefit, especially in a busy lab when you want to be doing things in different spaces and things like annotation of data, as I mentioned before. Maybe you don't want to sit there and label everything in front of the instrument. That's okay, because you can do it in a couple of different places and still get all that information recorded and not have to spend as much time on it. All right. One of the things that I really like here as well, and I mentioned this as far with regards to predicting things, you can, when you build your experiments, especially online, if people have used spectrum viewers and things like that in the past, you are able to get with these full spectrum instruments and with these standardized configurations, you're able to get a predicted value for each of these combinations and how well the kind of the prediction thinks they're going to work. But at the same time, you then take that and run it on the machine and you get the actual values out from the machine. So not everything works exactly the same way on every single day. So you can get that actual practical value out. And then that can give you an idea of where your trouble spots might be and where you might want to make an adjustment in your unmixing or in your controls or even in the fluorochromes themselves. So I personally think that's a huge benefit, because we do a lot of work in the front end trying to plan experiments and pick panels and make our best guesses of how things are going to work. This machine does a good job of doing that prediction, but also gives you the numbers to kind of verify how things worked for you on the other side. And if you can't see these numbers very well, I don't think. These dark blue numbers here are where it thinks there's a little bit of trouble. If you look at them as compared to on the theoretical side or the predicted side, the numbers are really close. The color matching is very good here, but the predicted versus the actual is actually extremely close on this machine. Or for that example, that's a 14 color panel that we did in the past. The longitudinal stability, I mentioned this already, obviously. One of the things is their physical characteristics. They use APDs instead of PMTs, and these APDs coupled with flat top lasers, which give you a wider beam profile and a much more uniform beam profile than some of the older technologies. This is becoming more common in new technologies, but than some of the old stuff that we had. Gives you a very nice uniform excitation, and then those APDs give you really good linearity over a large range of gains or voltages. The machine comes with the Cytec assay settings. That's the built-in normalization that it can do. I don't know if anybody was around way back when we were using application-specific settings on the FAXDiva software. FAXDiva attempted to do a thing where it automatically adjusted for you, but the machines were never linear enough. They were never stable enough, and it never really worked. But it actually does on these machines, meaning that once your unmixing is established and you're not making any changes within your panel, then you can maintain that unmixing, not have to rerun every control every single time because of that longitudinal stability. Again, nothing is perfect. It is going to change eventually. And if you make changes within your experiment, it is certainly going to change. But while things are stable, you can often get away without having to re-unmix every single time. And if you do, it's usually one or two parameters that you're having to rerun and not the entire thing starting from zero. And I think closing in on the end of what I wanted to talk about. If you have experience with the older technology, another thing is there's a lot of similarities with how things are set up in the software, such that if you're moving from the FAXDiva software to the full spectrum, it's called spectral flow, is what the software called. It's a pretty easy transition for people who are comfortable in the FAXDiva software. There's a very limited gain adjustments or voltage adjustments that you need to make. So again, with the traditional machines, we were often adjusting voltages and balancing things and making sure things are on scale. That becomes much less of a burden on these machines. And then again, once it's done, you don't have to redo it every single time because of the normalization. How much more do I want to say? Oh yeah, I won't go too much more at this. The whole offline access thing down here at the bottom. Again, you can build experiments in the cloud for free and practice and see how things work and make sure you're comfortable with it before you sit down in front of the machine and actually are spending money and using real samples. You can get a little bit more comfortable with it. So that, again, with respect to the match configurations, that can be really useful. The weaknesses, I mentioned this already, but that theory thing, it is a different way of thinking about approaching the experimental planning and stuff. Making sure you get the proper controls. Again, Tim's going to mention this a little bit more. But in the end, it's still flow cytometry. It's not that much different. You're still going to prepare the samples in more or less exactly the same way. Your FCS files are going to come out in exactly the same way. The fluorochromes you use are exactly the same. So there's not that much difference, but there is a little bit of difference in the theory and stuff like that. And then the record keeping, because of that longitudinal stability, it's easy to kind of get a false sense of security and just assume everything's going to work every single time. And we know that's not quite true. So you do have to be maintaining good records and making sure that you're maintaining your lot consistency and things of that nature. When everything is run as a one-off experiment, that's kind of built into it. But now it's easy to get kind of lulled and miss things occasionally. And that is, I think, about where I want to finish. So again, I think if you haven't already, this is Sweetums, by the way. Sweetums is one of our 5 laser instruments. If you haven't already, I recommend you giving it a shot. The technology is, as far as I'm concerned, the new thing. And I want to encourage people to try it. And that goes for the new machine as well. That goes for the A8 as well. We have not gotten our feet under us especially yet, but I do expect that it will have a lot of the same benefits, especially with regards to panel design and flexibility and stuff like that. Okay, I'm going to stop here. I don't know how long I lasted, but anyway, I'm going to stop here. Oh yeah, of course. Yes, please. Yeah.
The 8 has a ridiculously high rate that they say they can analyze.
Yes.
If you don't do imaging, it's 35,000.
Yeah.
So what is the Cytec rated at?
I think their rate that they say is about the same. I personally am comfortable.
Like,
Yeah, no, I'm personally comfortable for the Auroras between 6,000 and 8,000 without any difficulty at all. If you want to go much further than that, I start to get a little bit nervous, but you probably could. I don't know what you guys are.
different speeds.
No, they're certainly not orders of magnitude or anything like that. Yeah, if it's a couple thousand events difference, that might be true, but I wouldn't expect so.
About the number of detectors you're
Yep.
lasers and detectors. There's actually it's the same detectors across a bunch of different lasers. You're not dividing. You don't have 64 different emission spectrum that you're analyzing.
That is correct. So the UV laser covers from 370 to 850 or so.
Right.
And then the violet laser comes from 400 to 850. So there is some overlap there.
Just kind of trying to compare the two machines because they're different companies.
Yes.
The spectrum, let's say, just take PE, let's take the green laser.
Yep.
How many different places do you analyze off of that in the A8 versus the Cytec?
I don't know what the number is in the A8. With Cytec, it's 16. It's 16 chunks. So there's a difference in philosophy between the two companies here. Cytec has gone with very uniform, very kind of rigid breakdowns. I think it's 15 nanometers for almost every detector, and that's regardless of where it falls. It's just 15, 15, 15. Whereas the new machine, the A8, they actually kind of fine-tuned it a little bit. So you'll have one that's 20, and then you'll have one that's 10, and then so they did that because of kind of optimizing for specific fluorochromes, right? PE is PE. Everybody knows it, right? So why not have one optimized for PE and then have some other stuff around it? So I don't have all the specifics, but there is definitely a different philosophy there. How that impacts side by side, we don't know yet. Yeah, but that's going to be exciting. I'm looking forward to knowing, but I don't know yet. Yeah. All right.
One last question.
Oh yeah, of course.
So is it the A8, the new one is meant for people who want to do experiment with the imaging, or can you just run as a regular full spectrum?
That is a great point. It does have imaging. You do not have to, if you want to use it as a full spectrum instrument and just plow through some samples, that's perfectly fine as well. I would encourage you, if you're going to use the A8, to try the imaging side of things, especially get some right field images, right? Just because why not? But I also expect it to be able to be a workhorse instrument for people as well, right? We only have 2 Auroras. They are quite busy. So I'm telling people to try them, and then they're going to look at the calendar and be like, what? Anyway, they are quite busy. I'm hoping that the A8 can alleviate some of that stress. But again, we're not quite there yet. I'm also 100% happy if people want to start arguing to administration about buying a new Aurora. That'd be great too. So keep that in the back of your minds. Okay.
Yep.
I forgot I didn't even use this, but I do have a lid.
Good. Thanks. Yes. Sure, I got the right point. Okay. Thanks, Matt. So I was just going to go through what I will talk about, and also one thing I won't talk about. I would like to talk about panel design because we spent a lot of time on that software, but I thought these might be better for a short discussion today. But happy to talk about the panel design some other time. So I thought I'd go into the unmixing, and one of the questions people often have is beads versus cells to develop single stains, because getting those spectra is important to get them exactly right. So then compensation, the relationship between unmixing and compensation is worth a discussion, I think. And then I wanted to shift gears and talk about clustering analysis for high parameter data, which of course is what you get out of the spectral machine. And a particular offshoot of that I want to spend a little more time on is a tool that we built for batch registration. And that is based on our clustering algorithm. And it's pretty effective. It actually worked better than we thought it might. And it's a freestanding tool in the sense that you can take files done in different batches on different days, register them so they're more similar to each other, and then it simply writes FCS files so you can analyze them any way you like. So it's a nice freestanding tool for that. So if I get to it. So where am I going here? So First of all, I'm going to go reasonably quickly. Matt's introduced the machine and sort of how it works. And I didn't want to try and give a whole explanation leading up to this point. So please ask if I've gone a little too fast and left out too many steps. But to develop the spectra that Matt was talking about that are going to be used in the unmixing analysis, there's a lot of discussion about how to do single stains, whether you do that on capture beads that capture your different reagents, or whether you use actual cells and have the antibodies bind in the regular sort of way. And it's an interesting discussion because the beads obviously are very convenient. You can use them for any fluor antibody combination. And we can build some software that is very quick for developing the spectra from those. It works a lot better than the photo version. And so beads are great. But the second advantage they have is that if you're looking at very rare populations like IL-17 positive cells, it's such a tiny population that if you try to stain cells with that reagent, it's very difficult in a single stain to cleanly isolate that positive population and get a good spectrum from it. So that's a difficulty for using cells for everything. So again, the advantage is towards these. So on the other hand, cells are more realistic, and I put that in quotation marks because it's not, and unless this recent developments I don't know about, it's not quite clear what all goes into determining these spectra. There are subtle differences between beads and cells, but one I think of that might be significant is that when you look at reagent molecules that actually bind to antigens, so in other words, using cells, those are going to be pretty well-behaved antibody molecules. They're not denatured very much because they've still got their full binding ability to bind to the antigen in the cells. On the other hand, if you use beads, the anti-Ig beads, then I think your antibody could probably be a lot more denatured and messed up antibody binding on the cells. And you might imagine that the spectrum of those two kinds of spectrum, more perfect, less perfect, might be quite safe. But regardless, there's been lots of discussion. And I know last time I heard from Cytometry, I think they were saying, yeah, sometimes beads are better, sometimes cells are better. So it's but I wanted to show you how much it matters. And to do that, I just want to go back to this point about the mixing and compensation. So starting off, you have your, on Cytec, the 64 channel cross horizontally here, and you have all of your different fluors. So each one has a full spectrum, and that's the unmixing matrix that you feed into the calculation to separate out all of your actual fluorescence colors. And so that reduces your 64 channel data down to 30 colors, 32, whatever you have in your particular panel. So once you've done that, you can still then go and do some compensation by the traditional mechanism that you would do if you were using one of the older style machines where one color was given one shade. And it turns out that unmixing is rarely perfect. I haven't been able to get it perfect. And I think it's true for a lot of people. It theoretically should work, but there's always some little bits and pieces in there that aren't quite right. And so it's possible to fix those using a compensation matrix after you've done the unmixing. And I'm not sure of the mathematics of that. We use it because it works, but I'm not sure it's, my guess is a mathematician might not like that particular sequence of events, but it works. So for the compensation matrix, we can do the standard thing, is the Flojo version of the matrix editor on a fairly small panel, and you simply dial in the numbers you want to modify the data to clean it up. And you can see, for example, here, these two channels should not be, these two markers should not be correlated. In fact, they're showing a correlation. It's great. But the idea would be to do as much as you possibly can with the most perfect unmixing matrix you can, because you don't want to spend a lot of time doing the correction. So I've got a little comparison of beads and, so first of all, to evaluate where you are after the unmixing. So this is 20 antibodies here. But you can look at the cross correlation plots of the n by n plot of every parameter against every other parameter. And that's a pretty nice way to find when something's gone wrong. I pointed out this one a bit earlier. That one I think I already fixed. There's a couple of others that might need some help, but this is actually not a bad set of correlation matrices. So we had a panel we're developing, starting with a core of 20 specificities. And we were concerned about the speed cell issue, struggling a little bit with some of the mixing. And so we compared two possibilities. One is we do all our single stains on beads. And the other possibility is 16 of the single stains on beads and just 4 of them in cells. So I'll show you why that actually matters. And so first of all, here's the correlations. So if we look at the spectrum, every dot here is one of the 64 channels. And so if the beads and cells were given the same spectrum, everything would be nicely on the line. And you can see that they're a little off the line. And this is for person P, this is for BV 496. They're a little off the line. So does that matter? So this is now looking at, we're not applying any compensation, we've just done the unmixing. And we're now looking at correlations of various markers. So here we're looking at CD45RA versus CD8. I'll say right up front, CD8 is the bad guy in this panel. It often is because it's extremely high brightness. And you can see that if we look at all the bead unmixing, then you can see that there's some negative data here that we don't like. This one also is bending off towards the negative. This one is bending to positive. And in fact, CD39 versus CD8, it shouldn't be. Biologically, we know that that should be. So that is correct as well. And then autofluorescence has horrible interactions. So then if we look at the beads, 16 beads and 4 cells, so those subtle changes I showed you in the spectra, then we've lost the negatives here already. We've lost this negative. We've still got this problem here. And this is not perfect, but it's a lot better than the very strong effect. So the cells in this case, and I think what's striking is that the difference between the right and the left is quite strong. And that
All cells look like?
I'm sorry,
Well, what did to compare the all beads fairly to say whether beads are better than cells, you should compare it to all cells. And do the all cells look
we haven't done
better
all
than
cells yet. Partly because of this issue about the very rare ones.
Yeah.
So I think we're going to end up doing, it'd be nice to do the cells when we have beautiful signals like CD4, CD8, all of these markers where you've got a strong staining, clear positives and negatives. Those are easy to do.
I also think if you're dealing with an abundant cell, because it's going to measure the unstained as the bulk fluorescence of the bulk population. And if your cell's a small subset and has a different autofluorescent profile,
Absolutely.
then your unstained is going to be totally inappropriate.
Yep, which is another major issue.
But for some of them, like, it's good to see this actually, because I got a couple problematic cells I might want to throw in.
Yes, I think at this point I'd be tempted to say, yeah, cells might be a little better, but if you try to push, well, IL-17 is my favorite example where you put 1 million cells in and get maybe 10 or 20 cells that you can work with. And I think the Cytec won't even let you use 10 or 20. Yeah, we do the unmixing outside Cytec, but still, it's hard. So then if we do the compensation, so this second row is now compensation where I've tried to optimize for the bead samples. So I want all of the panels on the left to now look better. I don't care about what's happening on the right. And you can see that I was able to get rid of this little tongue going negative there. So this one actually now looks much more like this one. We can get rid of this negative data. This one we've got back down in line where it should be. And we've got a reasonable improvement. There's still a bit of problems there, but it's a lot better than it was for the autofluorescence. On the other hand, you can see that now we've introduced more problems on this side. So that it's not that we should be doing that compensation uniformly for this particular panel. It depends very much on the exact single stains that you're using. And of course, we did the other one as well. And this is now optimizing for the right. And you can see that, so we didn't have to do very much, but we could certainly straighten out this particular one here. And the changes I made, this panel I'm reasonably happy with. The changes here were for the 4 markers, 4% minus 1.5, 2% minus 0.7. So those are pretty small tweaks in the compensation matrix to sit over on top of the So I have spent a lot of time trying to work with an automated version of changing the actual the actual unmixing matrix, which would be a preferred way of tuning this. And it's still, that's a difficult problem. Okay, so I was going to talk a little bit about, we're moving, time's going by. How about if I take for granted that we have a nice clustering algorithm, and I'm going to go straight to one of the particular uses of it. So first of all, when you have high parameter data, you could spend a lot of time on one sample and try to analyze all the possibilities, but really it's not really feasible for a person to look at all the possibilities. And part of the problem is because some populations, you can't really define them just by bivariate gating. They're mixed into each other in such a way that to really separate them, it has to be a multidimensional separation between them. And so the computer can do that, we can't. So I think you have to go towards the algorithmic analysis for the high parameter data. And we developed, quite a long time ago now, we developed with Gaurav Sharma a clustering algorithm that has a lot of sensitivity for rare populations. And it turns out that was because those are the samples we used to develop the algorithm. So we got what we asked for. But then we realized that this gave us a cluster map of our samples where we knew in high dimensional space where all the different clusters were, how many cells there were, the breadth of the cluster. And so it was a very nice map describing the sample. And we thought maybe we could use that to register samples, because even with the Cytec, and I agree very much with Matt, it's a very stable instrument in many ways, but it's not perfect. And I think probably the bigger problem is that the people in the lab aren't perfect and the reagents aren't perfect. So if a reagent is fading over a few months, or if you have to shift batches unexpectedly, or if the person's protocols aren't as tight as they should be, which in research labs is common, then you will get batch effects. And so if you go over weeks and months, then the batches are distinguishable from each other. And so we were able to use our cluster information to basically lock together each batch to a sort of single super sample and then say, what does it take to make the clusters, each cluster match the other sample? And we take all the vectors that we derive that way, and we apply them then to the individual samples. So we're trying to keep all the biological variation between, different biological groups in the data. We just want to get rid of the batch effect. So there's those two kinds of variation normally, but we're trying to basically take out one kind of variation and keep the other. And it works surprisingly well. So my last slide, but it's a complicated one. So this is, I think, my favorite demonstration so far of the registration algorithm working. And this is data from Fred Hutchinson, where it was a large HIV vaccine study. It was about 5,000 samples. And these are UMAP plots using all of our cluster information to try and decide how are these samples all related to each other. And in the first panel, the batches are color-coded. And you can immediately see that you have serious batch effects. Each color tends to be a little patch. And you'll notice these ones over here, these were done 11 months later, and it's very hard to keep everything constant for that time. So not surprising, although the guy who did this, he was annoyed when they told him he had batch effects, but he's okay with it now. So what you really want is to have the samples in different batches all sort of more mixed together. And so we applied our registration algorithm to this data, and the second panel is what we got. Now UMAP simply mixes all the batches up and says that really, in contrast to here, where the batch variation is the dominant variation, now we have a situation where the batch effect is not really that strong in separating out the cell populations. And to show you that gives you some pretty precise information, you'll notice that there's lots of little microclusters in this data. So on the third panel, each color now represents one person. And these are samples stimulated with different reagents. And very importantly, they are blood samples taken over a period of about a year. And yet after registration, then we find that one person tends to associate very closely with the samples from that same person. And it's an interesting argument that people have that signature where you can tell the difference between them with quite a lot of precision. You know, the fact that, so all of these samples that were over here compared to here, basically you see the two halves here. That's basically the batches from here and the batches from here, but they've been brought together as individuals. So it's a strong registration method. And I think it also tells you something interesting about setting up clinical studies where people are very consistent with themselves. They don't change very fast. They have that recognizable signature in their blood cell populations. Okay, so I went through fast on some things there. There may be questions before I hand over to Nate.
Are you currently in the practice of always expecting to do compensation post unmixing? And if you don't have to, you're happy about that, but you're expecting that it's probably there.
So I am expecting that it'll be there. I would say that data I showed you from Chris Hearn, it was fairly recent. I would call that pretty good compared to some of our other panels, which were not so happy. So, you know, putting in 1 or 2% of compensation in a few channels, that I would consider great. We've done much worse. And in fact, if you look at some of the OMIP data that's been published, if you dig into the actual data, they've left in some of there's some fairly strong interactions that are still in the data.
Right. Yeah.
They're by no means straight and straight.
Right.
Yes.
I'm not quite understand why that rightmost panel UMAP there is actually those cluster like a individual participants are separate like that. Wouldn't you expect them to mix? I don't know what the cluster parameter based on.
So the UMAP is driven by all of the cluster information from the panel that we're using. So there's many different clusters, and the number of cells in each cluster, all of that is taken as information for the dimensionality reduction. So then you can say, well, which samples are most like each other? And it turns out, and we've seen this in pretty much every data set we've looked at, that when you add in all of those different clusters of different cell types, people do have a fairly consistent signature. And you can see it, the last two plots are actually the same plot, just with different coloring. So this plot and this plot are identical except for colors. And you can actually see here, for example, you see there's the pink and yellow samples with a microcluster here. When you look at the colors being individuals, they're all red, it's one person. And so these two different batches have been brought together Over here, the pink ones are here, the yellow ones are here. So it's brought together. So let me phrase it this way, that if we reduce the variation of the data due to batch effects, it allows you to see other kinds of variation. And here the variation is very much purpose. That makes sense. The fast. All right. Ready? Let's turn over to Nate to reinforce what you can do with algorithmic processing.
So we're up against time and I won't hold it against you if you want to leave, but I'll try to move through quickly. Do I just close out this one completely? All right, so I've been here forever, since 2002. No, 2004. My first job, I was running 4 colors on a fax caliber. I kind of miss those days of the simplicity of just 4 colors. But since being at U of R, I started by operating a cell sorter. Then I moved to doing big data projects on the LSR2, rest in peace animal. Spent hundreds of hours on animal doing conventional flow. And then a large project doing Cytoth. And now I'm doing full spectrum on the Cytrack Aurora. So I've had my hands in all these technologies along the way. But for the first big data project with the conventional flow, a little note here, I became an expert at crashing FlowJo. It just wasn't scalable to the need that I wanted to, based on 15 colors then was a bit complex, but now it's relatively low parameter. But back then, because I had hundreds of SCS files, I needed something a bit more reproducible and scalable. So that's why I moved into an R-based workflow. So what I'm going to show today is a piece of my project that's been piped through my analysis workflow. I will advance the slide. All right.
Whatever.
And so I'm showing you a practical application of what we talked about today. So not so much the technicality of unmixing and all that, but just actual primary data of a panel that was established earlier this year. This is a cohort of human participants that they were vaccinated with a primary series and then they received a booster. And so then we're measuring their PBMC at day 1, 15, and 90 post-booster and looking for antigen-specific CD4 T cells. And this is in a 33 color panel where the primary focus is mainly cytokines, but we also have an exploratory component where we're looking at tissue homing markers, which I won't show today, but I'm just highlighting the added complexity of high parameter data. You can have your basic phenotyping backbone and then add on to that experimental markers of interest. And all the data I'm showing now it was processed and analyzed in an automated fashion. Scalings are derived from the data, clusters, everything is done automatically or semi-automatically without ever having to draw a gate in FlowJo. And that keeps it scalable and allows me to process batches as they're generated. And more importantly, the derivatives that are generated are human readable. So we can operate in high dimensional space, 33 dimensions. You can't actually look at 33 dimensions. So plots are just traditional 2D. This is just a typical plate setup. In our project, we actually, per these blocks, we collapse them down or pool them using live cell barcoding, and I'll show that in a few slides. So each of these blocks are processed as one single super sample, and that allows for reduced reagent consumption and a much more uniform staining because it's a single tube. So this is primary data directly post-acquisition for batch 10. So this was run July 3rd of this year. And what I'm showing are the 6 primary markers we use for Is that showing? Yeah. The 6 primary markers we use to identify immune cell subsets. So we have five major phenotyping markers and viability. That then is passed into Flowsum, and it generates immune cell subset clusters. And if you rank order those clusters, your most proportional cluster of PBMC is going to be your CD4s. And what I'm showing here highlighted is the primary data that is assigned to the CD4 cluster laid over the aggregate data in black. So you can see it does capture CD4 in a way that you wouldn't gate it. So you do get kind of these spurious events, but that's not, that if you were to gate in FlowJo, you'd have hardline boundaries. Here, it's just data driven. And so for the sake of robustness, there's a certain amount of fine tuning you can do, but you're just going to let the data do the driving, so to speak. And then this is just another way of looking at the cluster assignment. The Flowsum nodes themselves embedded in UMAP space, highlighted in black, are the CD4 nodes. And if you wanted to further analyze this, you could find CD8s, gamma deltas, monocytes. You could find those immune cell subsets in here. But for this project, we were mainly focused on CD4s and CD8s, but I'm not going to show that. Then after we capture the CD4s comes the my favorite part, because it just looks pretty. These are 7 CD45 barcodes, and the way they're barcoded, that each sample will have a unique combination of 3. This is shown in aggregate, so you see all these collinear associations. But if you do it high dimensionally, you actually get, in this batch, there were 30 barcodes. You get 30 of these pots of cells. And then at the top here, PowerPoint said that it thought it was a group of leaves on a gray background. I thought that it was interesting, but it does present itself as data as art. So this is just, you don't want to see interconnected or overlaying barcodes. So this is the good result. And so then you've collapsed your pool, you've barcode assigned, and then this is just another QC report where you see that association of the unique barcode. So then you can recover sample-specific FCS events. And then this goes on into the meat of the analysis, where you want to get a readout of your analysis or your assay. In this case, peptide-stimulated T cells. So we have unstim on the top, nucleocapsid in the middle, and the spike wild-type peptide pool on the bottom row. So you can see in this particular donor, you have a qualitative response at spike that seems to go up at visit 51, which is the cohort encoding for day 15, and visit 49 would be day 1. So you can capture this information. Again, this is operating high dimensional, in a high dimensional manner. I'm just looking at 2 dimensions because that's what my brain can handle. And then the end, I didn't show the final Flowsum cluster where you're identifying all cytokine positive cells. So that's TNF, IL-2, and interferon gamma. And so based on this data-driven analysis, you can capture the kinetic of the boost, where you have expansion of these T cells, and then they mostly contract by day 90. And so this is just for one single batch. We're up to 15, 2 blocks per plate. So the effort of coding this out allows for, a robust and reproducible script where I can relatively quickly generate these numbers. And then just a quick analysis summary. So usually with a project, there's the dreaded deliverable that you need to pass on to someone else. In this case, we were contracted to report the number of antigen-specific T cells. To get to that, I used 18 dimensions. And then that leaves another 15, which is the exploratory component. So in a full spectrum panel, this is probably where you're going to spend the most time. It has the most complexity because you're down to a discrete immune cell subset, CD4s or CD8s, and then you have an additional 15 markers. So in our case, some combination of chemokines and tissue homing. And so how those are associating in a circulating population in the blood, that just takes a lot of time and annotation, a lot of heat maps and cluster plots. And so if you're new to full spectrum or you're expanding a conventional panel, you can think about it in two ways. A direct question that can be easily answered just in one single experiment. And then as you accrue and build this longitudinal data set, how can you leverage all that data to kind of really explore and do a deep dive and hopefully discover some nuance of an immune population that hasn't been published on before. And that ends my spiel.
Are you using your automated or semi-automated analyses for all of those steps, or you're using that to break it down to a manageable chunk that you can actually deal with? Then, are you're hoping to do all of it? Still developing the second part.
So I build in interventions just because of if something is off, if it's not actually capturing CD4s, then that's like 20 minutes of just rerunning that just to get it to fine tune a parameter. But for the most part, the way it's been working is I could probably just hit run on my scripts and come back, and this would all be generated and captured. I don't want to give up complete control to the machine, so to speak. So I like looking at the primary data. Sometimes you leave lab, your brain's mush because you're trying to think about things high-dimensionally. So if you have something, we've all worked in this space, right? These bivariate plots. And so seeing that the QC output just to confirm that things are copacetic, that's what I prefer.
I think that, for a lot of the pipelines, really like your semi-supervised pipeline, because you can set up something very beautiful to process one data set with one set of questions. But almost invariably, if you change the question, you're going to fine tune the exact analysis. And I think there's quite a lot of choices in your pipeline that are specific to this project.
Yeah, so obviously it's tailor-made to your project, but you can generalize this to large panels. The most important thing is your naming conventions, right? As long as you consistently name things with expected characteristics, then you can auto-generate the metadata, auto-generate the AXI labels and things like that. You can certainly all do this manually in Flojo, but for me, it was a frustrating experience as the complexity of these panels grew, as the longitudinal data grew. When you're sitting on half a terabyte of FCS data, having to redo everything in Flojo is just a nightmare.
Is there a way of ranking your output with a question that you want to answer is how far has it gone along a continuum? So you're not moving from here to here to here to here to here to here. You have a factor that just changes over time or changes after treatment, and it's a continuum. There are no discrete boundaries. It's a continuum. How does the readout of the change of a cell along a continuum occur in this system?
So I think that's more the exploratory component where some of those markers are heterogeneous. So like for the cytokines where they're on or off, it's really easy to capture that information, essentially, especially if they associate high dimensionally. And sometimes with these panels, what you think is an interesting cluster could be a spectral artifact. So I think there's no real ideal way to like a one-stop shop for analysis. Like everything has to be tailored to your particular project need.
So I think it's sort of a general problem that a lot of these cluster algorithms, they find clusters. and they don't find ridges that connect, you know, if you think of the mountain.
Or pathways, like Monocle will look for a pathway and it's not concerned about separate spots.
Right, but that's
Right? It's
a
a pathway, and a lot of times what we're looking for is movement along. a-
Yeah, so yeah,
kind of.
Yeah, I don't know whether you can do the same algorithm.
yeah, if you have the proper longitudinal sampling of your, like, of a developmental trajectory or a course over time, this, along the way, this generates this derivative FCS files. So you start with your source data, it spits out your CD4s, it spits out your sample specific FCS files. And so you have that record of data, and you can always just kind of create a module to plug in at whatever step where you want to do a pathway analysis.
It'd be nice to have a pathway analysis, a way of having that work into, so there's splitters and there's pathway people. Like every time, like you're either like you got 5 cells, so let's make 6 clusters. Or you've got 5 cells and you have to rank them in an order, or it's a Y versus a W, you've got to define a tree, right? And as biologists, both of those are perfectly legit things to do. But if you're not an immunologist, the tree thing becomes very important. So if we move out of human immunology and try to make flow available for a lot of other systems, what's going to be helpful is to be able to somehow get ranking along a continuum.
Yeah,
I
right.
think Monocle is nice, but I think there's a huge number of algorithms now available for trajectory analysis. And I think it's a tough problem.
It is.
Because often, you know, the problem is if certain steps in a differentiation pathway are rapid and some are slow, then it's going to be hard to recognize some of those connections. But I think it's now a very active area. I don't know what the people's favorite algorithms are, but I know somebody who they looked into them years ago and they found 50 of them that
For
were.
flow, there's not a lot of options for pathways. Gemstone, which I don't think is still being made, was one of the only ones.
That was, yeah.
There are
Assumed linear.
Right, well, because it's like you doing your correction where you know what the thing is.
Yeah.
Like we know it's linear, so that's an assumption we can make.
think like Sony and Cytobank, they have nice tools, but they're locked behind proprietary software. This is R-based and, you know, philosophically that's open source. So anyone could take my code if they want. Wouldn't recommend it, but it's a really niche audience that actually wants to look at the code and see what it's doing. But I have put quite a bit of effort into it, and it can be generalized to other data sets. So it is available for anyone interested.
Nate, wearing one of your other hats with the development T-cell work with Kristen. How much have you gotten into the pathway or trajectory analysis on those?
Not so much. So I want to go back and reanalyze it in this fashion. Like so like every project I learn a bit more. So I can revisit that mass cytometry data because I don't think that's been fully mined. We also have what I haven't tried yet is merging the single cell by mass cytometry or full spectrum with the 10x genomic sequencing. That's like a higher order of analysis that I don't know if I have the brain for that, but because the dimensionality of the genomic data, that's like, you know, thousands of parameters. So
I'm sorry. Go ahead. Yeah, I was thinking particularly for the neonatal samples that may be time when things are changing relatively rapidly. So that may be more like a hematopoietic sort of system where you have more intermediate stages and you're looking towards the trajectories.
yeah, so I guess I didn't do a shout out. Kristen Scheible, and as a neonatologist, she started her lab where we everything was a one-shot deal. We could run the sample once, that was it. We could never go back to try something else. From the pediatric samples, especially the babies, where it's a heel stick, you can get maybe a mil of blood. And so these high parameter panels are ideal for that. I know she's building a new cohort where it's more intensive sampling after birth, like through the first few weeks of life. So I think she has that in mind. It's a multi-omics approach, single cell, proteomics, metabolomics. I don't know what she has in mind for merging those all together.
Single cells and added like a site-seq panel. So you have antibody barcoded and the single cells in one shot. So you can do flow, but I think those might be a better option if you want protein and genomic on the same
Right.
I
side.
also think when we start blending in some just simple microscopy, cell size, nuclear size, and shape, which are all parameters we're going to be getting in.
On the AA.
We'll actually get off the image stream is probably going to give you more accurate ones because you can comp and get more than like you can only use about 3 floors for imaging.
Right.
You can't comp it with any of the other things, so it's very limited. You get compensated with multiple stains, so this type of high analysis. But you know, those change on a different scale, but they could be worked in here. It'd be cool.
Yeah, obviously the you know, it's becomes exponential the more parameters you add.
You can narrow it down to like
Yeah.
maybe 3 or 4.
Well, there's a certain irony for how I approach things because I always like you want to operate high dimensionally, but I got to break it out into discrete workable chunks, like just focus on phenotype or just focus on cytokine or just focus on tissue homing. It's when you throw everything together, you can spend months on that one heat map, figuring out what cluster is what. Is it biological? Is it just a high dimensional artifact? But it's a fun challenge.
And I know we're already over, but I want to come all the way back if you would. And this slide brings it up. You were a bit of a difficult convert to go from the mass cytometry to the full spectrum. How are you feeling now? Honest answer. You can tell me.
Well, if throughput were not an issue, I would do CyTOF every time. Yeah, with CyTOF because it's time of flight, and unless you somehow change the fundamental properties of the universe, those isotopes are always going to fly through the chamber at the same time. So you don't have any spectral issues at all. You can just, in a week, order antibodies and run a panel. There's very little in the way of optimization. For my CyTOF project, we were running 500 events per second, getting cloud fusions. Ideally, we should have been around 200 or 300 events per second. On the Aurora, I can run 6,000 events per second. So the throughput is just unmatched with a flow-based system versus the injection system of the CyTOF. But I think the data quality is just better in the CyTOF. It's if I could just run them more, like more of them. I don't know with the new instruments, or is that just a physical limitation?
Yeah, I don't think that rate's ever going to really go up, unfortunately.
Yeah, it's such a pity because that's the only thing that holds us back, really.
Yeah.
Otherwise, it's lovely data to work with.
Right. You can't run 78 parameters on an A8. You can't run 64 parameters on a SciTech. There's always some sort of concession. You have to like live with weird unmixing or having to do compensation.
All right. Clear now, but so thank you to my speakers, to Ms. Lee, to everybody for coming. As always, please do bring questions if you've got them. We are always happy to answer those and have those conversations. Yeah, thank you.